Assalamualaikum my dear aimers, hope so you all are doing well. Today the topic of our discussion is chemiosmosis. For this topic I would suggest you to first take the lecture of cyclic and non-cyclic photophosphorylation and then try to understand this topic. So let's start today's topic. Firstly, chemiosmosis is a combination of two words. You see chemi and osmosis. Chemi means chemical and the chemical over here is H positive ion or a proton. And osmosis means the movement of water molecules from high concentration to low concentration with the help of a membrane or a cell membrane or a semi-permeable membrane. But over here, osmosis means the movement of H positive ions. Okay, osmosis is the movement of H positive ions, and osmosis is always from high concentration to low concentration. And the second point about osmosis is that it is always with the help of a membrane. You know what's the difference between cell membrane and plasma membrane? Cell membrane is the membrane of a cell, while plasma membrane is the membrane of an organelle. Okay, the structure of cell membrane and plasma membrane is entirely same. Okay, so the membrane we are talking about in chemiosmosis is basically thylakoid membrane. So chemiosmosis will be the movement of H positive ions from high concentration to low concentration with the help of a thylakoid membrane. So the simplest definition of chemiosmosis can be this mechanism of H positive ions pumping across the thylakoid membrane. And you all know that what is the purpose of this chemiosmosis? The main purpose of chemiosmosis is to produce ATP because it's the mechanism by which ATP is produced. Photophosphorylation is the production of ATP and the mechanism by which ATP is produced in photophosphorylation is called as chemiosmosis. Now there is one more term which has been mentioned in your book, which is gradient. Gradient means to set up a difference of concentrations. At one place there is high concentration and at one other place there is low concentration. And whenever the things move from high concentration to low concentration, like when electrons move from high energy level to low energy level, they emit energy. Similarly, when the H positive ions will move from their high energy level to the low energy level, they will release an energy which will be used to make ATP. So this is the general concept about chemiosmosis. Now we'll look into the details and the most important points for chemiosmosis. So first point about chemiosmosis is that it takes place in thylakoid membrane because cyclic and non-cyclic photophosphorylation takes place in thylakoid membrane. The second point is the product of chemiosmosis is always ATP. Third point is the chemiosmosis take place in both cyclic and non-cyclic photophosphorylation because the purpose of cyclic phosphorylation was to produce ATP and the mechanism for ATP production would be chemiosmosis. The next thing is the structures which are involved in the production of ATP. So basically two structures are involved. First structure is the electron transport chain of photosystem 2. Okay, plastoquinone, cytochrome complex and plastocyanin. It is involved in the synthesis of ATP and the second most important structure is ATP synthase. You know H positive is an ion. An ion cannot cross the membrane. You know that the membrane, this thylakoid membrane like that of cell membrane is made up of phospholipids bilayer. It has also proteins, channel proteins and carrier proteins. Now what happens that these H positive ions cannot cross this phospholipid bilayer. They are transported with the help of a protein. Now which protein? Channel proteins. Channel proteins are those which give a path or a channel to the ion to cross through it. Okay, so suppose these are the channel proteins and this channel protein gives a path to cross the H positive ions, and with the help of these proteins the ions move into the thylakoid interior space, or you may say into the inner side of the membrane. So this ATP synthase, as the name says, ASE, so ASE means enzyme. So enzymes are proteins. So this ATP synthase basically is a channel protein which provides path for the movement of H positive ions from thylakoid, from the outer side of thylakoid to the inner side of the thylakoid. Got it? And one more important point, that this chemiosmosis is a coupling reaction. Coupling means it is coupled with some structure. And the structure with which chemiosmosis is coupled is electron transport chain. Like chemiosmosis cannot occur without electron transport chain, plastocyanin, plastoquinone and cytochromes complex. Got it? Students, now we will try to understand the process of chemiosmosis with the help of a simple diagram. So here you can see that this is a thylakoid diagram. The white portion over here is a membrane of thylakoid, which is exactly similar to that of cell membrane. So here you can imagine that there is a lipid of phospholipid, there is a layer of phospholipids and channel proteins and all that stuff. And the inner space of thylakoid, the matrix of a thylakoid, is called as thylakoid lumen or thylakoid interior space, while the matrix of chloroplast is called as stroma. So fluid outside the thylakoid is stroma and fluid inside the thylakoid is lumen or thylakoid interior space. Got it? Now there are some structures associated with the thylakoid membrane. You all know that those structures are firstly photosystem 2, then comes plastoquinone, then there are 2 cytochromes which are iron containing proteins, then there is a plastocyanin, copper containing protein, then photosystem 1, and then ferredoxin, and then NADP reductase. Do remember that all the photosystems and its components are embedded in the thylakoid membrane, while NADP reductase is present on the outer side of the thylakoid membrane. You can see that I have made it out of the thylakoid membrane, on the outer side of thylakoid membrane. Okay. Now what are the structures present inside the lumen? The structure inside the lumen is water splitting enzyme. And do remember that this water splitting enzyme is present on the inner side or inner surface of the thylakoid membrane. Got it? Now, there is also one important stuff over here, that is ATP synthase. This ATP synthase is that channel protein which will help in the transport of H positive ions. Got it? So, this is the whole structure which is involved in the process of chemiosmosis. Now, what happens that sunlight energy falls on photosystem 2. Electrons of chlorophyll A of reaction center of photosystem 2 accepts that light energy and the electrons are excited. These electrons then travel to plastoquinone and then the electron hole is created in photosystem 2. To fill that electron hole, photolysis takes place in lumen. This water breaks into 2 components, hydrogen and oxygen. This hydrogen further splits into its components, H positive ions and 2 electrons. And these 2 electrons from water fill the electron hole in the photosystem 2. I have explained all this process in non-cyclic photophosphorylation. Now, what happens next? That this process of photolysis continue to take place in lumen, because I told you that non-cyclic photophosphorylation is a continuous process. It occurs continuously inside the cell. So, photolysis also occurs continuously inside the cell, and the concentration of H positive ions continue to increase inside the lumen. So, you see that there are many H positive ions in the lumen. Now, you all know that this stroma is a storehouse for many chemicals. Enzymes like Robesco and many other chemicals are present in stroma. Similarly, H positive ions are also present in stroma. But the concentration of H positive ions in stroma is less than the concentration of H positive ions in lumen. Got it? Now, we saw in the very first slide that for the process of chemiosmosis, we have to set up a gradient. So, the concentration of H positive ions in lumen should be much greater than the concentration in stroma. So, for that, what would we do? We will transport these H positive ions from stroma into the lumen. As the transport is from low concentration to high concentration, so the transport will be called as active transport. And you all know that for active transport, we need energy. From where does this energy come? This energy comes from the excited electrons from the photosystem 2. Now, what happens that when these electrons come on the low energy level, okay, like the cytochrome complex and then plastocyanin, when they reach plastocyanin, then these electrons release all of the energy that was absorbed by them. Okay, this is the same amount of energy which was absorbed by the electrons. Now, this energy of electrons is used by cytochrome complex to transport these H positive ions from stroma into the lumen in this way. Okay, so this cytochrome complex is called as proton pumper because it pumps wherever the word is written pump, it means it's active transport. So this cytochrome complex pumps H positive ions from stroma to lumen. Got it? Now the concentration of H positive ions in lumen is much greater than that in the stroma. Now students, what happens next is that these H positive ions are now present in lumen. Then all the energy which was present in the electrons is now transferred to H positive ions, is now stored into H positive ions. And what is the stored energy called as? The stored energy is called as potential energy. That's why we say that these H positive ions are now source of potential energy which they got from the excited electrons. Now, These H positive ions pass through this ATP synthase because H positive ions cannot cross the phospholipid bilayer. They go through the channel protein, which is ATP synthase. And as the lumen is in high concentration with H positive ions, stroma is in low concentration. H positive ions move from high to low concentration by the release of all the energy that was stored in them. And the released energy is now used to combine inorganic phosphate with ADP to make ATP. In this way, ATP is synthesized. Now, the same happens with photosystem 1. The energy is accepted by the electrons of photosystem 1. The electrons reach to ferredoxin and the electron hole is filled by the plastocyanin. From ferredoxin, they go to NADP reductase. Now, NADP reductase has got the electrons from photosystem 1 and H positive ions which are now present in stroma are also transported to NADP reductase. Now, NADP reductase combines the NADP plus present in stroma, 2 electrons from photosystem 1 and 2 hydrogen ions or proton from water to make NADPH plus H positive. So do remember this equation, and I have explained this equation in the previous lectures. You can go and check out that lectures too. So this is the process by which ATP is synthesized. Now do remember two most important points. Light reactions, cyclic and non-cyclic photophosphorylation and chemiosmosis, they all take place on thylakoid membrane. But the product, which is NADPH and ATP, they are released in stroma. You see that this whole black portion is stroma. So the NADPH is basically produced or released into stroma, and this ATP is also produced or released into stroma. This was the question which appeared in last year Mdcat, that where are NADPH and ATP formed? So the options were thylakoid membrane and stroma. So the answer is stroma because ATP and NADPH are produced into the stroma, while light reactions take place in thylakoid membrane, chemiosmosis takes place in thylakoid membrane, but the products are then released into the stroma. So this is the MCQs you have to remember. Now, this is the diagram which has been mentioned in your book. This is somewhat complex. You can just make it simple by making the boxes over here as I told you in the previous diagram. Okay. So you see that this white portion is thylakoid interior space or lumen. Then comes thylakoid membrane. You see the structure is exactly same to that of cell membrane. And this pink portion is stroma. Now look here that photolysis is taking place in lumen. This pink portion is stroma mentioned over here. Like you can see from here. This is stroma. And then these are NADPH and ATP. So you can see that NADPH and ATP, they both are synthesized in stroma. So it has been mentioned in your book. And the one more point you have to remember that this ATP synthase is a channel protein. Got it? Now, I have mentioned the points of a whole process. Okay. So the point is firstly the definition of chemiosmosis, which is the explained form of chemiosmosis, the process of ATP synthesis using free energy, and free energy is obtained from electrons which are passed through several carriers of electron transport chain is known as chemiosmosis. And then you have to remember the sites where the reaction is taking place. Photolysis takes place in lumen. As photolysis is taking place in lumen, so active transport is from cytochrome complex, and this cytochrome complex is called as proton pumper. Now, active transport takes place from stroma to lumen, and proton pumping is across the thylakoid membrane. Proton pumping means the pumping of H positive ions, and as the word is pump, that means it's an active transport. Now, passive transport takes place from lumen to stroma with the help of ATP synthase. And do remember one point, that the process of chemiosmosis is basically the passive transport of H positive ions from lumen to stroma. Okay. This is basically the point from where the chemiosmosis takes place. So chemiosmosis is a type of a diffusion. Do remember this, as osmosis is also diffusion. So chemiosmosis is also a type of diffusion. Now, what is the summary of light reactions? Do remember these points. First is breakdown of water. Photolysis takes place in lumen. Oxygen is released in lumen. Light reactions take place on thylakoid membrane. Photosystems are located on thylakoid membrane. ATP is produced or released in stroma, and NADPH is also produced in stroma. Got it? Now, what to do next is that this is the processing time, and I have assigned you some tasks to do. Don't make your face like that, okay? You have to complete the task, and the task is that you have to practice the diagram of chemiosmosis at least 2 times. The diagram which was from your book, I also mentioned in the slides. So do practice it 2 times, and if you, when you will be done with the practice, then do inform me in the comment section on Instagram, and then your face must be like this. Okay, so do practice it. Now means now. Okay, so practice it. So this was all from my side. I hope that you have got the lecture, and if you have got the lecture, have a big smile on your face. Be happy, make others happy. Like the video, share it with your friends, and subscribe to my YouTube channel. See you in the next video. Allah Hafiz.